ELISA was a technique which was conceptualized and developed by Peter Perlman and Eva Engvall at Stockholm University in Sweden. Enzyme-linked immunosorbent assay utilizes an enzyme system to show specific combination of an antigen with an antibody. So, in simple terms, if you want to understand this, it is a system in which this is an antigen, it is capable of binding with an antibody, and as soon as it binds with the antibody, this antibody contains some enzyme. There will be one more antigen which will come and bind over here, and as a result there will be expression of a color. The color can be blue or it can be yellow. Now, depending on the amount of color which is developed, we can determine that what kind of antigen or hormone or enzyme is present in the solution. So with this idea we begin. It is a method of quantifying an antigen immobilized on a solid surface. The solid surface is a microtiter well. They are around 96 to 120 to 30 microtiter wells in which we place all the antibodies, and then after antibodies laden with the enzyme, and it varies for different kind of reactions, different kind of proportionality is there. Eliza uses a specific antibody with a covalently coupled enzyme. This is very important, which I tried to show. See, this is an antibody, suppose. It will have a position where an enzyme is present. This is the enzyme which is attached with the antibody. The amount of antibody that binds the antigen is proportional to the amount of antigen present. No extra amount of antibody will be present because we keep on washing it with tris phosphate. The enzyme system of ELISA consists of an enzyme which is labeled by a specific antibody or antigen and a chromogenic substrate. Chromogenic means color providing substrate, which is added after the antigen antibody reaction has taken place. So I said that one of the antibody will bind over here. Sorry, I just said the reverse. This is the antigen, antibody is the cup in which yes. So after this entire process is done, then after there comes the addition of the next antibody, and which gives the chromogenic substance. It means the color is developed only after that. The substrate is hydrolyzed by the enzyme attached to the antigen antibody complex, and this results in the color development. An ELISA test uses components of the immune system. That's why I said IgG, which is immunoglobulin. Immunosorbent, that term comes from the usage of this immune system substances like IgG or IgM, which are antibodies, and some chemicals from the detection of immune responses in the body. And this ELISA test, it involves an enzyme. It also involves an antigen or an antibody, or both of them, and it is used in order to diagnose various diseases, sometimes food allergic reactions, and most of the times it is done for pregnancy tests. HIV test is identified with the help of the same immunosorbent assay, enzyme linked immunosorbent assay. The antigen antibody reaction, chromogenic substrate specific to the enzyme, O-phenyl dihydrochloride or for peroxidase, or para-nitrophenyl phosphate or alkaline phosphatases are added. Why? Because they are the substances which help in formation of color or development of the color. The substrate is acted upon, or it is hydrolyzed by the enzyme attached to the antigen antibody complex to give a color change. The color in the reaction can be read visually, or it can be detected with the optical density calorimetry. Calorimeter is a kind of device in which we can place two test tubes, and one can be used as a test, and whereas the another one there will be some difference in the level of absorption of light, and we can determine that what is the color which is developed over there, and is it a positive change or is the antigen or antibody or the hormone or enzyme present in the microtiter well or not. That can be determined with this calorimetric color change. The antigen or antibody is coated on a solid surface such as plastic tube or well or microtiter plate. These are certain plastic wells in which we can let the antigen antibody reaction occur. And once the reaction has occurred, now all the extra antibodies they need to be washed off, and for that we use tris phosphate to clear extra amount of antigen or antibody which is present in the microtiter well. Enzyme immunoassays can be used for detection of either antigens or antibodies in serum and other body fluids of the patient. The antigen or antibody labeled with enzymes are used, and alkaline phosphatase, horseradish peroxidase, and beta galactosidase are enzymes which are used in the enzyme linked immunosorbent assay tests. There are different types of ELISAs which are available for detection of quantitation, for detection as well as quantitation of either the antigen or antibodies in a serum or body fluid. There are basically four types of ELISA test. First one is direct ELISA, and you can see over here. At first, there is an antigen which is attached to the substrate. The black line can be taken up as a microtiter wells base. So at first antigen is added, then after antibody is added, and this primary antibody is added with the enzyme. That pink color substance is the enzyme. As soon as the substrate is added, the color change can be detected. Let's see. Antigen is attached to the polystyrene plate. That is what I said. Enzyme label antibody is added that can react with the antigen and a substrate that can be measured. Second is indirect ELISA. In this, antigen is attached to a polystyrene plate. Addition of primary antibody followed by an enzyme labeled antibody is done that can react with both the primary antibody and substrate. You can see at first antigen is attached, then after first antibody, the green antibody is the first antibody. In the second phase, second time what we do is we add secondary antibody, and this secondary antibody is a conjugate because it is bound with the enzyme. Now all the antigen and primary antibody will react with the secondary antibody and form a structure which is a conjugate structure containing the antigen, primary antibody, secondary antibody along with the enzyme. And as soon as the substrate is added, again there will be color change which can be seen. Now in the sandwich ELISA it is little different. You can see at first the antibody is there. Let's see. A capture antibody is attached to the polystyrene plate. So at first an antibody is placed over the microtiter well, then antigen is added that specifically attaches or captures the antigen. Second antibody, also specific to the antigen, but not to the same capture antibody, is added, and the sandwich is done in with the antigen. So antigen is in between two antibodies. The second antibody is then followed by an enzyme labeled antibody specific for the second antibody that can react with the substrate and can be measured. See, at first we are adding an antibody, then the antigen. After the antigen, secondary antibody. When primary and secondary antibodies are added, the antigen is sandwiched between two of the antibodies, the red and the green one. Later on, we add the antibody which is linked with the enzyme, and it has the capability of linking with the green antibody. And as a result, this entire conjugate is formed, and as soon as the substrate is added, the color change can be witnessed. What about the next type of ELISA? It is computative ELISA. Generally, whenever we are performing any of the ELISA, we or the most sensitive kind of ELISA, it is the sandwich ELISA. And for HIV test, we use the same technique. Computative ELISA. This test is like the sandwich ELISA, but involves the addition of computative antibodies or proteins with the second antibody when the second antibody is added. This results in a decrease in the substrate signal that is generated. This test can be done considered to give good, highly specific results. At first, we are adding an antigen, then an antibody, which is having the presence of enzyme. After that, we are adding competitive ELISA because we will be adding the inhibitor antigen as well. So as soon as we add the inhibitor antigen along with the antibody, we find that there is no coupling taking place between both of them, and as soon as the substrate is added, the color change is developed. So this is a technique which is used widely in order to determine the presence of an antigen or an hormone or enzyme in a body or in an organism. And we also try to understand that what kind of sensitivity each of these ELISA has. Generally, sandwich and competitive ELISA are practiced much more than the direct and indirect ELISA, but any of the processes in uses the same idea, that is, enzyme is linked with the antibody which is immunosorbent. It means whenever we talk about adsorbent, it means it is just attached to the microtiter well, or it is attached to the substrate. So I hope you all have understood.